We are announcing a new world model for protein biology, a scientific engine that can predict, design, and discover proteins. It is built on the latest evolutionary scale modeling, or ESM, foundation. The engine learns from billions of protein sequences that evolution has generated, and it uses that knowledge to represent proteins, map them, predict their structures, and design new proteins, all the way from atomic interactions to evolutionary relationships spanning billions of years. Three components make up the system: ESMC, ESMFold2, and the ESM Atlas. ESMC is a state-of-the-art protein language model. It was trained on billions of sequences and learns the rules of protein biology. Compared to its predecessor, ESM2, ESMC pushes a new scaling frontier, delivering stronger performance as the model grows larger, especially in understanding long-range structural relationships. ESMFold2 builds on the 6 billion parameter ESMC model. It is a cutting-edge structure prediction system that has been validated for designing protein-protein interactions. In benchmark tests on the FoldBench dataset, ESMFold2 outperforms other models in the doc pass rate for both protein-protein and antibody-antigen complexes. It also offers a dramatic speedup when used in single-sequence mode, thanks to an efficient diffusion sampling algorithm. In the laboratory, ESMFold2 has been tested on 5 therapeutic targets. By inverting the model, feeding in a target sequence and searching for the best binders, researchers can generate de novo mini-binders and antibody-derived single-chain variable fragments, or SCFVs, that show high hit rates, nanomolar binding affinities, target specificity, and functional activity. A notebook is coming that walks through the entire design loop, from a target sequence to ranked binder candidates, and the full protocol is detailed in a preprint. ESM Atlas is a map of 6.8 billion proteins that covers the full breadth of life's biodiversity. Thanks to ESMFold2's massive folding throughput, more than 1 billion predicted structures can be included. The atlas is organized according to ESMC's internal world model. To make that world model interpretable, the team trained sparse autoencoders, an unsupervised neural network technique that decomposes complex representations into a sparse set of roughly 16,000 interpretable features. Each feature is described in natural language by an automated pipeline that links it to known biology from protein databases. Collections of these autoencoders, trained on different model scales and layers, are released for researchers to explore the functional relationships that ESMC has learned. If you want to experiment with the models on your own machine, the code is available on GitHub. For example, installing the package from the repository lets you run ESMC locally with a simple Python script, import the model, load the tokenizer, and pass a protein sequence to the model to obtain hidden states. If you prefer not to set up everything yourself, the Biohub platform offers a straightforward API that handles authentication, encoding, and inference behind the scenes. Using ESMC through the Biohub platform is even simpler. After installing the ESM Python package, you import the client library, create an object that represents a protein sequence, and then call the encode method to get an embedding. The platform also gives you logits or embedding outputs directly. The same approach applies to ESMFold2 on the platform. A client object named capital SEQUENCE capital STRUCTURE capital FORGE capital INFERENC is configured with your API token. Then you build a structure prediction input containing protein, DNA, or ligand sequences. And finally you invoke the fold-all-atom method to get a predicted structure, complete with quality metrics like PLDDT, PTM, and ITM. These results can then be written out to a CIF file for visualization. The team has also released sparse autoencoders for ESMC. These are implemented in PyTorch. Load the base ESM model and the autoencoder weights, initialize the specified layers, and run the model on a sequence. The output includes sparse activation tensors that identify which latent features are active for a given input. The embedded natural language descriptions of each feature can be consulted to interpret what functional aspects the model captured. Beyond the technical details, Biohub has established a dedicated safety team that assesses potential biosafety and biosecurity risks before release. They evaluate each component, including ESMC, ESMFold2, the Cs, the Atlas, and the Binder-Distance system, to devise safeguards. As a result, all model weights, code, and data for the core components are released under a permissive MIT license. The Atlas dataset and the Binder-Distance workflow are also openly available, so the community can reproduce, extend, and scrutinize the work. For those who use these models, The creators ask that you cite their recent preprint on the language-emital world map of protein biology, as well as the supporting papers on ESM3 and the open source code repository. The main citation follows. Candido, Salvatore and colleagues, 2026, Language Modeling Materializes a World Model of Protein Biology, preprint, available at Biohub AI Papers ESM Protein PDF. Similarly, the ESM3 paper in the code repository should be referenced with the provided citation details. All software is licensed under MIT, so any derivative work can build on this foundation.